Advanced Medicine Coalition, Procter & Gamble, Robinson, Bradshaw & Henson, or RBH, Roche, Schwartz Communications, Theragen, Third Rock, Exconomy, and Unilever. We are all very grateful to all their help in many ways. Furthermore, a very special thanks to the Genomes, Environments, and Traits Steering Committee, Karen, Daniel, Mikhail, and Jason. I want you to see their faces so you see them around. You can talk to them. And we have special guests here from local high schools and a very important contribution from PG Ed, Ting Wu and Dana Waring. I'm going to conduct this kind of an interview of myself. We're going to have a few interviews coming up. It's going to be interesting. So I'm going to ask frequently asked questions, not to preempt, but to whet your appetite for what's coming up. And even though we barely started this meeting, there are already people asking, when's the next meeting, okay? And the history of this meeting was in 2006, my wife and my daughter and I, and my daughter will be here, you'll see her, went on a tour of Toronto, Ann Arbor, New York, and some other places for people who had contacted us about the Personal Genome Project that had just appeared in Scientific American, and it's probably why they were contacting us. And we wanted to see what people want, and that's sort of the tone that we're setting here today is what do we want? What do we want to see in the future? What are we seeing already? From the front lines, getting together the first 13 or so people that have their sequence. And then we've done this every year. We've gotten together the volunteers in the Personal Genome Project, and this year we thought we'd do something special by aligning it with DNA Day, which honors Jim Watson's contribution both to the structure of DNA and to starting this genome project. And then, and Jim was kind enough to join us on this day. And we hope to do this in the future. In addition, there's a Consumer Genetics Conference coming up on June the 2nd, and the third annual Personal Genomes Project at Cold Spring Harbor. So you can see Cold Spring Harbor figures prominently in this. People have already asked me what's the favorite part of today's agenda. Well, I haven't been through it yet, so I'm not sure, but I would have to say imagination, imagining the future. I've heard a lot of very imaginative things already, and we will be honoring some of these imaginative ideas from the trenches at the very end of the day with awards from Ian Torrent and Nomi, and hopefully you will be inspired by the awardees in this definitely grassroots movement. So what happened on DNA Day? April 25th, 1953 was this publication in Nature that some of you have heard of. 2003, we declared victory on the Human Genome Project for the third time out of many. And in 2008, a very important act was passed unanimously after 13 years of non-unanimous non-passing. This is the Genetic Information Non-Discrimination Act, which deals with health insurance and employment. It's important. So we've got this incredible success of the DNA structure. However, about half people get the DNA structure backwards in mirror image. Jim and Francis got it correct, amazingly. And we haven't finished a single human genome yet, I am sorry to say. There's about 7% of it that's missing from every genome so far. And you could say sour grapes. We don't care about that, that part. Pay no attention to the genome behind the curtain. But just as an example, on chromosome 1 there in the upper left is 20 million base pairs that are missing, and there's a very likely candidate allele for multiple sclerosis sitting in there that's we having trouble mapping for the last 5 years because of incompletion. This entire set is over 200 million base pairs, which is enough to fit in almost all of the model organism genomes from the beginning of the Genome Project into that gap. Second somber note is that pharmaceutical is rising. Does this mean that healthcare costs will rise too? Maybe, but one ray of hope is that the cost of sequencing is dropping exponentially. Not exactly clear how you connect the dots, but that's what we'll be talking about today. Everybody wants to know who is my choice for the technology winner. These are 20 of my favorite companies. These are radically different from the sequencing technology that we used to sequence the first human genome, and they're radically different from each other. And I find them all very inspiring, and I hope there will be multiple winners. So the answer is yes. So what happened in 2005? We had this lovely, slow exponential curve that looked a lot like Moore's law for electronics playing out in DNA sequencing. We had basically a monopoly, no offense, a very good one that served us well. But in 2005, we got this abundance of new ideas in sequencing and the end of the race, which was a bit of a distraction in my opinion. And now the price has plummeted from $3 billion, sort of at the culmination of 2000, 2004, to $1,500 cost of consumables as of the end of 2009, and it's come down a little bit since then. Now I'm going to claim that we're getting close to the $0 genome. What do I mean by this? Wikipedia, internet searches, maps, and translation, these are basically free. They cost somebody something, but they cost the consumer nothing. Here's the building that we're in, provided by Bing. I, of course, favored Microsoft in this illustration. But what does zero mean? We have the cost is getting down around $1,000, but the price is not $1,000. We're still paying off research and development. We're paying off venture capital. We're doing a lot of things. The price is around $20,000, or the price is $0. But what are the strings? Okay, if you're $0 if you're in the Personal Genome Project, but the string is you have to share your data. There's all these other possibilities of different business models where these businesses will recover your the $1,000 cost and give it to you for free, and you've just got to very carefully inspect the label on each of these things. And it's not just the one genome that you're getting, that you're inheriting from your mother and your father. There are going to be multiple genomes in your life. Your immune genome, your cancer genome will change from day to day, and your microbial environment will change. And that's what we mean by, that's one of the things we mean by environment. So there's a concern that if we get our genetic advice, it's going to be generic advice. Is this what's going to happen? If it's just going to say exercise, drink your milk, eat these good foods, get more rest. Unless sometimes even athletes fall dead from cardiac arrest if they have hypertrophic cardiomyopathy. You can have bad reaction to celiac disease, lactose intolerance, fava bean problems, cholesterol, and narcolepsy. Are we just cheerleading? Are we talking only about celebrating the dropping of prices and growing value? Again, I'd like to embrace imagination here. We should imagine the unintended consequences, both positive and negative. It's beyond our thoughts if we don't think about it. We're not claiming that we all have to share. We're celebrating people who are sharing, but there's almost 7 billion of us. Just 100,000 or so would make a big improvement in our ability to do research. Here's a controversial one. What if the government starts testing babies for intelligence genes? Well, they already do. Nearly all newborns, at least in the United States and similar countries, are tested each year for PKU. This is a wonderful example of what we're talking about here today. It's 100% genetic. If your child is homozygous for this, they will be mentally retarded, 100%. If you change their nutrition, however, they will be normal intelligence, whatever the range is. 100% environmental. It doesn't make any sense to call this 50% environmental. It's 100%. You can change it. And from that standpoint, what should we do about genetic diversity? I'm not talking about ancestral diversity. I'm talking about true genetic phenotypic differences. The kids that are really sleepy in class or the kids that are really jumping around in class. Do we use drugs to move them towards the middle of the bell curve, or do we actually embrace some level of genetic diversity? Now, there are genuine diseases to be solved here, but there are these trade-offs. The question is, are we looking for rare alleles or common? The fact is that rare alleles aren't so rare. Each one might be 1 in 100,000, 1 in 10,000. You add them up, 10% of us have a rare disease, and most of us are carriers for one of those rare diseases. We have that rare disease in our family. And we're not talking about weak effects and so forth. We're talking about almost 1,800 genes are highly predictive and medically actionable, and this is recognized at the NIH and genetest.org. Not very few of these are in direct-to-consumer tests, but they're all available to medical geneticists. And this is not merely a patent issue. And you can see this number is rising here. You can see it's reaching up past 18. It's gone up from about 100 in 1993. What if there are no cures or no preventions? Do we not want to know about it personally, much less share it? Well, I think I was inspired long ago by many of these individuals. These weren't necessarily experts in their field. Even Doug Melton, who's a scientist at Harvard, he was a frog biologist when I met him. Augusto Doni, of course, was not a lipid biochemist, and so on. But each of them said that their family was important and other families like them was important, so they want to search, raise consciousness, raise money, and search for cures for all these different diseases, some of which interact in various ways and are not separate. So just to drive this point home, why public gene plus trait plus environment data sets? Why not make it all private and encrypted? There are barriers to research. There's barriers to serendipity. And I think that one could argue that some of the experts, Feynman made a contribution to O-ring analysis, even though that was not his field. And Wally Gilbert, who was my mentor, was a meson physicist. And he made a pretty big contribution to chemistry and biology. And I guess Jim was an ornithologist and phage biologist, not a crystallographer, but he made a huge contribution to crystallography. So we need to embrace non-experts, and families are probably the best non-experts to embrace. And here's a recent example of the Madsen family. And people ask, how many genomes do we need to sequence? And it's amazing what you can get out of 4 genomes in one family. 2 discoveries of 2 very important diseases, one affecting the lungs and another one affecting both the face and the limbs, happened in this family, and sequencing the 4 of them resulted in discovery of both genes. Another issue is about re-identification. We can promise our participants that the data will never escape, and if it escapes, it'll never be re-identified, but that would be wrong. That would be disingenuous. It sometimes escapes, quite frequently, and we felt that it's safer to tell research subjects that this is quite likely, and you should consider very seriously whether you want to have it in the open. But nevertheless, there are multiple attempts to encrypt, and then there are follow-up studies that show the encryption didn't work, and this will go on. And what about cell lines? That's a big deal. You might be able to encrypt the data, but it's very hard to encrypt the cell lines. They have the ability to make a copy of you now if you possess the cell lines. Does that mean nobody gets access to the cell lines? So we have this unique project, the Personal Genome Project, which does make sure that the participants know what they're getting into. They have to get 100% on an entrance exam. And the numbers here are not their prison numbers, but where their cell lines are stored in Coriell, to whom we're also very indebted. And this is intended not to be studying one disease at a time, but getting large cohorts where we can study multiple diseases that might interact in various ways and might interact with other traits which are not classically considered disease traits. We have IRB approval for 100,000 centered in Boston, but we have now various branches around the world, each of which can have their own goals. We have 16,000 volunteers already so far, and we're aiming at this diverse set of traits, and I'll show you just a few examples going on here. How does PGP differ from other projects? There's the Human Genome Project, the HapMap, 1,000 Genome Projects. There are no traits in these, and I think this is a pretty obvious missing information. dbGaP and genome-wide association studies, there's no sequence. For the most part, that's changing, and it's not public. That's not changing. So this is a frequently asked question. How many complete public genomes are there? Putting aside the fact that we're all missing 5 to 7%, I'd say there's about 13. In here you can see there's 3 families, but in each of these families, at most, one genome is truly public. The Madsen family has 0 that are public, even though they've disclosed 2 of the most stigmatizing, potentially, aspects of their genomes. How do we improve interpretation? Some people think that this is very far off in the future, and I would argue that it's actually close if we act as a community. Here's an example from PGP number 6. We found a hypertrophic cardiomyopathy allele in this individual. But at the time, there was only one example of this exact DNA base pair change in the literature on the far left, and then our unknown on the far right. But we knew that there was a lot of people storing this, a lot of physicians and companies storing this away in their filing cabinets, and so we asked around and find a lot more examples. And so to make this more automated and more curated, we've established evidence.personalgenomes.org that I encourage you to look at, where you can actually say what is the background level, can you actually do diagnosis on new alleles that crop up or people that are adopted, because the standard of practice is to do it only if you have a family history. This is kind of an overview of the different alleles that exist, and you can click on a particular one and find out what its properties are, or you can look at it from the standpoint of the person rather than the whole genome community. Here's our oldest PGP volunteer, number 11, which is Skip Gates' father. And this is a prioritized list of alleles found in this individual, and like many African Americans, he has the sickle cell trait, which can have health impacts much less than a homozygote. And you can drill down on each of these things, determining whether the allele is rare and what the literature is on it, and so forth. And so we encourage the community, and there's quite a number of people contributing to this way of looking at the evidence for individuals. Another question is, how many traits? How do we decide which traits? And we're not going to read through this slide, but I would say one way to look at it, and this is just one of many, is to think about it from the point of treatments, even very minor ones. These constitute large industries, vitamins, minor drugs, many of them over the counter are not even drugs at all. And I think from looking at this point, it helps us think about traits. But there are many ways of looking at traits. They don't have to be immediately life-threatening ones. Another aspect that people ask about is epigenetics or epigenomics. And in the Personal Genome Project, we're trying out a few things that may or may not be extendable to 100,000 individuals. But include their ability to take a small sample of skin from anywhere on their body and turn it into stem cells, and then turn those stem cells basically into any tissue. In the future, this could be used for personalized therapies. In the present, we're using it to study how their epigenetics works. And you can see here's PGP number 1 and PGP number 9 on the far left there, male and a female, reprogrammed from skin to all these different tissues. And you can see actually some similarities. For example, we see respiratory epithelium, as you might have in the lungs, the blood, the bone, neuro tissues, cardiac tissues, and liver tissues, among others, have been reconstituted. And we have many articles now that have come out of the Personal Genome Project and the stem cell projects. We're collaborating with a Harvard researcher, Randy Bruckner, whose group is correlating genetics with functional magnetic resonance imaging of the brain under various circumstances. And I think there's been at least 2 PG volunteers that have been through this so far, but that will crank up quite a bit. Randy has room for 3,000. Finally, there's this example of something that lies in between our environment and how our genome plays out, which is we are studying the immunome, their immunological response to microorganisms and to ourselves, autoimmunity. And this involves looking at the rearrangements of pieces of DNA that exist in the genome, plus pieces of DNA that are made from scratch, creating great diversity on a daily basis in your blood system. This is done in collaboration with Roche and 454. We're very grateful. And this has been done so far with a fairly small subset of the PGP volunteers looking at vaccination to hepatitis and to influenza. And this is kind of the textbook case where you have a very low level of the blue response to antigens before you get even exposed to the antigen. Then you get vaccination or other exposure, and you get a 1,000-fold increase. And then you get a booster there, and you get another factor of 1,000 for that antigen, but not for any others. And so we've been following this time series for 2 years now, and have some very interesting results that I hope you'll ask me about in the hallway here. Too technical for this particular moment, where I'm trying to set tone rather than report on all the results. But it really is exciting that we're finding connections between particular rearrangements in the immune system and the pathogens to which they bind. So, in summary, we have the personal genome. We don't have to go from the personal genome directly to the traits. We were trying to integrate these with intermediates like the epigenome and measurements in the environment based on what microorganisms you have, and Rob Knight will tell us more about that, and your immune response to them, and many other measures of your environment and your traits that we've talked about. So actually started a little late and finished a little bit early, so I think there's a little bit of time for questions if there are any. Yes, Stephen.
Your brand new article from research that just was published and handed out. There's a lot of interesting things in this article.
Yes. Jantine Lonsoff, who's in the audience here, and Margaret Huhe are responsible for this article that summarizes the Personal Genome Project. If you contact them or me, Stephen has brought it to our attention that this is a really up-to-date, just came off the presses on the Personal Genome Project and some of the ramifications of it. Yeah.
So obviously a huge emphasis on genomes. The tools are so powerful. Preston Hensley, and the science is romantic. But to understand biology, it seems like we have to match a deep understanding or a deeper understanding of the connection between genotype and phenotype. And while, as you point out in your article, that's going to be difficult because there's many layers of phenotype and it's more complex, it seems that genotype is half of the story. And what are your thoughts, or anybody's thoughts, on how we're going to attack phenotype and then pair the two up to understand biology?
It's less than half of the story. I mean, it's in this conference, it's environment and traits. And all, in a certain sense, what we want to do is get past the point where we're saying this is your genome, your destiny, get used to it, to this is the various ways the environment has changed your genome impact. One family has the same alleles, has no effects. We want to find out what that family is doing right. It may or may not involve a new drug, may involve a lifestyle change. So the point requires that we share our traits and our genomes, at least some subset of us, so that we can find the correlations and either go through the causative scientific biological research or the correlative where we find an environmental change and test that in clinical trials or other trials. One question back here. Keith.
Keith Batchelder. George, as you talk about this, I'm reminded of the efforts to raise community around sharing information. Do you have any thoughts on that?
Well, I think we're seeing some community enthusiasm around this project, and we'd like to encourage it around any other projects like it. I mean, this is not intended to be a monopoly of any sort. It's really to try to encourage lots of different ways of thinking about how we can create community. The Wikipedia model I'm inspired by, and I think there's a similar thing happening in genomics right now. And again, not every company and every individual has to participate. All it takes is a few to really make it work, and I think that's what we're seeing at this meeting, and any suggestions are very welcome. Well, I think there's going to be plenty of time for additional discussion, but I want to keep us on schedule and turn this entirely over. I will not be directing it, turning this over to Robert Korwich and Carl Zimmer, who have really very graciously agreed, and I think went well into the midnight oil planning this morning's session. So I'm going to turn it completely over to them, and I get none of the credit, but if there is blame, I'll take it. Go ahead.
You can blame us.